Okay, everybody, we had you look at pages 19 through 25 today, and essentially that came down to looking at organelles, right? Kind of wrapping up where we were with organelles, what they do for the cell, getting into plant and animal cells and a little bit of differences between their organelles, and then moving into cell transport, which also has to do with certain organelles. So, a little review. We talked about this yesterday in class and in our video. The different organelles here, if you wanted to key out your interactive notebook of what the nucleus, vacuole, etc. here do and where they're found. Then cell membrane through centrioles, where they're found. And you can see most of these are found in both plant and animal cells. Some are found in only plant, some are found only in animals. More of them are found only in plants. Take a look there though. Okay, so what are some of the structures that allow a cell to perform its role, right, in an organism? And remember, cells are the smallest living unit, right? They can do without you, right? You can't do without them, lots of them, but they can do without you. And why? Because they're capable of um, essentially carrying out homeostasis on their own, and that's because largely of their organelles, right? So things like their nucleus and their cell membrane, right? Their cell membrane sets up a boundary, right, around them. It allows them to maintain an internal environment regardless of what's kind of going on around them. Okay, you probably know something here about the difference between plant and animal cells. One of the big things are plants can produce their own energy from the sunlight. We'll get into more details and more terminology about what that means, but ultimately, plants can feed themselves. All right, so if you look here, organelles in an animal cell, you're going to see a lot of similar ones in a plant cell, right? Because they're both eukaryotic cells, right? They both have a nucleus, they're both fairly large, they both have cell membranes, they both have mitochondria, they both have cytoplasm, right? They both have a lot of stuff in common, but a plant cell has a few extra things. So again, here I'm just going to put this up so you can key out your work. And if you look here, right, the matching, obviously, right, the cytoplasm, the jelly-like structure, etc., right? So you can key out your notebook so you have something to look at, study from. There's the animal cell and the plant cell again. So, what structures are present in an animal cell but not in a plant cell? And then what structures are present in a plant cell but not in an animal cell? So, what are the organelles that do each of these things? Chloroplasts are the reason that plant cells can make their own food. They also have a cell wall and a vacuole. And some other things here. Plastids store food or pigments. The mitochondria converts food into energy, and that one's found in both. Okay, here's a Venn diagram. You might have come up with similar things, right? You can see what does a plant cell have on the left there, an animal cell on the right, and then all the stuff in the middle, of course, is what they share. And you can see the list in the middle is bigger, right? By and large, these are both eukaryotic cells. They both have a lot of the same stuff. But a plant cell, plants don't have skeletons, so their cell wall has to act as their structure. They can't move, so they need a large vacuole to be able to store materials. Um, because they can't move, they can't get food, they can't capture food, so they have to be able to make their own. Therein lies the chloroplasts, right? And then a few things about them. Structurally, plant cells tend to be more rigid, rectangular. Animal cells, maybe a little more pliable, more round. All right. And again, here, just to key out some of the work we asked you to do. If you take a look here, you can check that work for yourself. Okay, then we had you start getting into cell transport. And it's important to recognize it doesn't matter the type of cell. In order to carry out homeostasis, a cell needs to be able to move materials from the outside to the inside of the cell, and it needs to be able to move materials from the inside to the outside. But it needs to be able to control that process largely in order to maintain homeostasis, right? You can't maintain an environment internally that's different than the environment externally if you don't have the ability to control what's coming in and out of the cell. So when we talk about cell transport, there are two major types. One is called passive transport, and one is called active transport. So there's two big headings, passive and active transport in cell transport. Passive simply means that the cell itself is not playing a role, right? Nature, the activity of nature, right? The fact that molecules are always moving is really what leads to passive transport, right? Unless you get down to 0 degrees Kelvin, which has never been reached on Earth, right? You got to be 273 degrees below zero Celsius to get to where there's no movement of matter, right? As long as matter is moving, it's going to move from where it's more highly concentrated to where there's less concentration, right? Imagine spraying some perfume or some cologne in a room, right? Originally, it's going to all kind of smell right where you sprayed it, but eventually it's going to spread around, right? That's diffusion. It's spreading around. So much of the way that things get across the cell membrane, things like oxygen and water and carbon dioxide, is simply based on diffusion, right? And that's a form of passive transport. So you watched a video on that, and here it's defined for you. Simple diffusion is just the idea that materials, atoms, really, molecules, small molecules, move from an area where there's a lot of them to where there's not as much, where there's a high concentration of them to where there's a low concentration of them. Kind of like sliding downhill, right? It's almost as natural as gravity in some regards. Facilitated diffusion is just another form of passive transport, and this really just comes down to the fact that some molecules have a hard time moving through the cell membrane. So you want to think of the cell membrane as kind of like, I always think of it as like a screen on a screen door or on your window, right? Think about what a screen lets in and what it doesn't, right? Small materials, right? Air can move right through a screen. The flies, the bugs, can't, right? The grid structure keeps them out. That's kind of how a cell membrane operates. That's why it's selectively permeable, right? It'll selects certain things to be able to come in and other things not, largely based on size of those things. Some charge too, but largely based on the size. So molecules like oxygen, because it's in O2 form, right, in the atmosphere, CO2, water, very small molecules can move relatively easily through the cell membrane. But some things are bigger, right? Glucose. And so you need to have channels inside the membrane to allow movement. So we'll spend some more time reviewing these topics, but some terms that are related to how to think about how water moves through a membrane, across a membrane. Diffusion of water is just given the term osmosis, right? So don't get confused. Osmosis is really just simple diffusion, but when you're specifically talking about water, and why do we talk about water? Why does it have its own name? Well, because of course water is very important to all kinds of cell activities. It's a universal solvent, right? So a lot of things dissolve in water, and with that, are able to move across the cell membrane because of it. So some terms that are related to this, and for now, what I want you to be thinking about is your root words, right? Hypo, hyper, and iso. What do those words mean, and how is it related here? A couple things to remember when you're talking about these solutions. This is very important. Number one, when you're defining a solution, you're talking about that solution that's surrounding the cell. You're not talking about the solution inside the cell, right? The cytoplasm is a solution, but we're not talking about that. We're essentially talking about the blood, because let's face it, most cells, certainly in humans, are going to be surrounded by blood. So how can we describe that solution? That's really what these terms come down to: hypo, hypertonic, isotonic. And then the second most important thing, right? The first most important thing is we're describing the solution around the cell. The second most important thing is that we're describing the amount of solute in that solution outside the cell as compared to inside the cell. So hypo means below. So what that really means is that we have a below level of solute in the solution surrounding the cell. So in other words, we're surrounding the cell with more pure water than is what is inside the cell. And because of that, water is going to do what? It's going to diffuse into the cell, because water is highly concentrated outside the cell. Imagine you just drank distilled water, and less concentrated inside the cell. So what's water going to do? It's going to move in. In the case of a hypertonic solution, hyper meaning above levels, again, we're describing the solution outside the cell. Hyper meaning above levels, we're describing the solute load in that solution surrounding the cell. So if we have above levels of solute outside the cell, that means we have a lower concentration of water in that solution outside the cell as compared to inside. And so the water tends to move out of the cell. This is a dehydrating scenario if you surround a cell with a hypertonic solution. Iso, it's fairly easy. Iso means equal. Isotonic solution means we've got some movement into the cell, some movement out of the cell, but essentially the concentrations of the solute in and out of the cell are the same. Okay, finally, we had you look at an activity that tested this, right? Putting eggs that have been peeled of their shell in different types of solutions, and what happened to the eggs, right? Three things. You got rid of the membrane. The eggs swelled in size, being placed in vinegar. Why? Right? Think about it. The vinegar is very low in um, solute, right? The egg shriveled when it was placed in corn syrup, right? So in this case, the material outside the cell is very high in solute. So what happens, right? Where's the water going to go? Okay. So these are some tough topics. Spend some time thinking about them. If you get a chance, look them up, practice with them a little bit. A lot of terminology here. Okay, we'll see you soon. Take care, everybody.